Good morning, everyone. I want to thank the organizers for this amazing event and also the opportunity for me to share some of our work. So my story today is about how our structural biology group at Amgen was able to use cryo-EM to understand the drug action on TRPC6. So I just recently submitted the manuscript, and this is the first time I present the work to an external audience. So this is something new and hopefully also interesting to you. TRPC6 is a non-selective calcium-permeable cation channel. Its activity is important for the physiological function of many cell types, and its malfunction, usually in the form of enhanced channel activity, has been associated with several cardiometabolic diseases, including FSGS and cardiac hypertrophy, and these two are the most well-characterized so far. So FSGS, or focal segmental glomerulosclerosis, is a type of kidney condition in which scar tissue develops on the kidney's filtering units called glomeruli, and proteins leak into the urine. It is a serious disease that could lead to kidney failure, at which stage the only treatment options would be kidney dialysis or transplant. Because FSGS mutations, because gain-of-function mutations in TRPC6 has been identified in FSGS patients, so it is possible that a small molecule inhibitor of TRPC6 could be used to treat FSGS. Cardiac hypertrophy is and the abnormal enlargement of the heart muscle resulting from the increases in the size of cardiomyocytes and also changes in other heart muscles components. And TRPC6 and its close homologue, TRPC3, their expression levels are usually very low, but they are upregulated in cardiac diseases. And there are evidence that by inhibiting TRPC3 and TRPC6 can attenuate hypertrophy signaling in cells, and suggesting that there is therapeutic potential in TRPC6 inhibition. However, so far there is no TRPC6 targeting drugs on the market. And we believe that structural information on the modulation of these TRPC6 channels could potentially facilitate drug discovery efforts in those areas. And as I will briefly introduce in the next two slides, the best approach to study the structure of TRPC6 would be cryo-EM. In just a few years since the resolution revolution of cryo-EM, it has profoundly changed the landscape of structural biology, especially for large protein complexes and integral membrane proteins that cannot be crystallized. We at Amgen are very excited about the recent developments in the cryo-EM field and has built our cryo-EM capability from scratch to add another tool for our structure-based drug design. Here is just a brief overview of the historical overview of cryo-EM structures of TRP channels. One of the major breakthroughs in the cryo-EM field is the determination of the TRPV1 channel in 2013, which showed us that it is possible to solve a membrane protein structure at atomic resolution. And by the end of 2017, cryo-EM structures for TRP channels has been available for 5 of the 6 subfamilies. The only subfamily that there is no structure is TRPC, and which was solved in 2018. At Amgen, in late 2016, we installed our own Talos F200 microscope for grid screening. And at around the same time, we also started collaboration with UMass Medical School for data collection on their Krios microscope. And solving the structure of TRPC6 was the top priority of our cryo-EM efforts at the time. Here is the challenge we face in the protein biochemistry of the TRPC6 channel. So for the first structure, we used amphipose which are amphipathic polymers that can solubilize membrane proteins without detergents. So this is the first structure we have ever seen with any TRPC channel. While we are excited about this low-resolution structure, we are also very disappointed that the transmembrane domain was not resolved. So later, we also got the structure of the TRPC6 Fab complex in detergents, and again found that there is no transmembrane domain. So next, we reconstitute the complex into lipid nanodiscs. And we were very hopeful that in the lipid environment of the nanodiscs, we would be able to see the transmembrane domain, but still it was not there. And finally, there was a TRPC6 structure published, and we found that they use two different detergents, Elman G and CHS. We have tested these two detergents before, but never in tandem. And also, they reconstituted the protein into nanodisc after detergent purification. So we followed a similar protocol and finally get a structure with both the transmembrane domain and the intracellular domain resolved. We took great lengths to resolve the transmembrane domain because this is the domain that our small molecule inhibitor binds to. So here is our first complete structure, and the transparent envelope marks where the transmembrane domain is. At 3.1 angstrom, we could build the model for most of the protein, some lipids, and most importantly, the small molecule antagonist. So TRPC6 is a homotetrameric protein with 4 identical subunits. On the intracellular side, you can see there is a coil sticking out from the bottom, and the anchoring repeats surround the coil. And the transmembrane domain of TRPC6 has a similar design as other TRP channels and voltage-gated ion channels with 6 transmembrane helices in each subunit, separated into 2 clusters. S1 to S4 are in the peripheral region. We call this voltage sensor-like domain because they are similar to voltage sensor, but TRPC6 is not gated by membrane potential. S5 and S6 are in the center. They form the channel pore. And in this movie, you could see the small molecule antagonist, which is colored red, is binding in the pocket in the voltage sensor-like domain. And from the intracellular side view, you could see that the antagonist is, the binding pocket of the antagonist is accessible to the intracellular solvent. And we find that the density fit very well with the small molecule. And the structure also allows us to visualize the atomic interactions with the protein. So we further verify the interactions in the fluorescence-based assay. So the small molecule antagonist is from a patent by Sanofi, and it is a highly potent small molecule antagonist with subnanomolar binding affinity. And we know that the primary amine is critical for the potency, and in the structure it shows that it's interacting with 2 negatively charged residues, one glutamate from S2 and one aspartate from S3. And mutate either of these residues almost completely abolish the inhibition. The antagonist is also 35-fold more selective over TRPC3. And when we compared the sequence alignment, we found that all the residues at the binding site are identical except for this arginine residue, which in the structure it shows that the arginine interacts with the piperidine group, forms a cation-pi interaction. And we propose that the lysine residue in TRPC3 at this position is not at the ideal position to form the cation-pi interaction. And also in the assay, we found that when we mutate this arginine into alanine, it dramatically reduced the potency. And there are significant conformational changes at the binding pocket of these 2 side chains. So So the histidine has two alternative conformation in the apo pocket, and one of the conformation would interfere with the binding. And in the apo pocket, the arginine would also interfere with the binding. So the two side chains need to move away from the binding site to allow the antagonist to bind. And as I will show in this morph, from the apo form to the antagonist occupied form. So our structure provides the first view of the antagonist binding pocket in TRPC6 in the voltage sensor-like domain. And this is different from the previously identified antagonist binding pocket, which is located at the subunit interface between S4 and SF5. And in our structure, it is actually occupied by a phospholipid. We don't know whether the lipid has any function on the whether it has any effect on the TRPC6 function, but the binding site overlaps with the antagonist, and also it seems that the presence of the lipid blocks the movement of S4 to S5 linker. Now, in order to better understand how our small molecule antagonists inhibit channel activity, we thought that it might be helpful to get another structure with a more activated conformation for comparison. And because many of the disease-related mutations actually increase channel activity, so first we mapped these disease-related mutants into our structure and see whether we can find any clues about how these mutations lead to increased channel activity. And we found that many of these mutations are located in the intracellular side, mostly at the interface between the anchoring repeats and the two helices in the C-terminus. So at interface 1, we found that there are 6 mutations. And in our assay, we found that many of these mutations actually increased channel activity. At interface 2, M132T is a disease-related mutation that increased channel activity. And we can see that the methionine actually form hydrophobic interactions with the leucine and valine. So we asked whether introducing mutations into the leucine and valine could also have an effect on the channel activity. And actually they did. Mutating both valine and leucine into threonine increased the maximum channel activity by 2-fold. So we decided that this double mutant could be a good start to pursue a more active form of TRPC6. Furthermore, pharmaceutical companies have a lot of small molecules that can sometimes help with structural studies. And in this case, we accidentally identified a small molecule agonist in our high throughput screening for antagonists. And this agonist is much more potent compared to OAG, which is a soluble analog of the native lipid agonist diacylglycerol. So far, there is no TRPC structure with an agonist, and previous efforts to solve the structure in complex with OAG have failed, probably because of the low binding affinity. And we reasoned that with our small molecule agonist, we probably can reveal the agonist binding site. And we determined the structure of the agonist bound TRPC6, and indeed, we found the binding pocket of the agonist, which is located at the subunit interface between S6 and the pore helix. And this agonist binding pocket was occupied by a lipid in the antagonist bound structure. And at 2.9 angstrom, we can easily fit the model for the agonist. We can see that the agonist is mostly surrounded by hydrophobic residues. And mutation of residues around this region, many of them actually lead to a non-functional channel. Except in these two cases, you can see that the channel can still be activated, but at a much lower potency. Because this binding pocket is located at the outer leaflet of the membrane bilayer, and also it was occupied by a lipid in the antagonist in the antagonist bound structure. So we thought about the possibility that whether this binding pocket is also the same pocket as the diacylglycerol. So we tested mutations at this pocket, and we found that a lot of them actually eliminated OAG response. So it is possible that this site is also responsible for diacylglycerol recognition. What's interesting here is that although V706A mutation can be still activated by our small molecule agonist, it's not active in response to OAG. And similarly, for E672, it could still be activated by our small molecule agonist, but not by OAG. So that suggests that these two residues probably are more important for diacylglycerol recognition than our small molecule agonist. One last point I want to make here is that in contrast to the large conformational changes of the side chains in the antagonist during antagonist binding, we found that there is not a significant conformational change in the side chains during the binding of the agonist. So we do an overlay of the antagonist bound structure and the agonist bound structure, and we see that it seems that the agonist tilt the S6, and which is also associated with the movement in other regions in the transmembrane domain. And as you will see in this morph from the antagonist bound structure to the agonist bound structure, in the side view, there are significant conformational changes in the S4 to S5 linker and also the trip helix. And in the intracellular view, you could see that there is a slight rotation of the S6 helix that seems to enlarge the pore. However, both of our structures actually have a closed gate at the S6. And the electrophysiology recording of TRPC6 channel shows that even in the presence of the agonist, the open probability is very low. And the open time is less than 1 millisecond. So that suggests that the closed state is much more stable than the open state. And that's probably why both our structures have the are in the closed state. So maybe we need other tools or other TRPC channel variants to get the open channel conformation. But that's beyond the scope of our study. So overall, our work with two structures showed these two novel drug modulation sites. So these two novel drug modulation sites. And in the next few slides, I will. Sorry. So in the next few slides, I will try to. I don't think, is it out of battery or? Hello? Okay. So, thank you. So in the next few slides, I will try to compare these two, the two modulation sites we identified with the other modulation sites which have already been identified in TRP channels. And you will see that there are very interesting similarities and differences in these modulation sites. So in general, we believe that these channels, open and close, is controlled by the gate at the S6 gate, at S6. And we think that, and we found that the ligands identified in TRP channels occupy 3 different sites to control the gate at S6. So the first site is the agonist is actually the active site of the menthol pocket in TRPM8 channel. And we found that our small molecule antagonist binds to this pocket, and also there is a small molecule antagonist bind to the TRPV6 channel. And the position of this one at the voltage sensor-like domain suggests that the modulators at this region probably act on the trip helix or also the S4 to S5 linker to control the gate. And the second modulation site is the active site of the vanilloid gated TRPV1 channel. And it is located at the inner leaflet of the membrane bilayer. And we could see that in the apo form of TRPV1, there is a lipid at this site. And also from yesterday's talk, we found that the antifungal drug binds to this pocket in TRPV5. Okay. And also, in our structure, we found that this site is occupied by lipids. And also, there is the antagonist binding structure has published in TRPC6. And the last site is the active site of diacylglycerol, and also our small molecule agonist. And this site is at the outer leaflet of the membrane bilayer. And we believe that this seems to be a very unique activation mechanism compared to TRPV and TRPM. So in conclusion, the two sites we identify with our structures could be targeted for future design of TRPC-mediated diseases. And at Amgen, we are very excited by the cryo-EM developments in the field, and also we will continue to expand our cryo-EM capability. So far, our cryo-EM efforts have impacted both small molecule programs and large molecule programs. And for the organizers, please pay attention to the last bullet point. Maybe I could get another talk next year. So I want to thank, I feel very grateful that I had the chance to work with different functions at Amgen. And I also want to thank Shing and other groups in our member for their guidance and support. And thank you for your attention.
Thanks, Yonghong. Great talk. We have time for some questions. If we have any from the audience, maybe I'll start whilst we get a mic here. So I think you described it as the resolution revolution, which I think was a great term, and we've seen some fantastic structural insights across some talks. But just trying to get a sense of, I think it took you like 2 years from when you embarked on this initial sort of goal. Your timeline, can you just give us a sense of like how long it really took to get to like this 3.1 angstrom resolution that you showed?
So I joined Amgen in late 2015, actually started the projects immediately. And at the time, we don't have any microscope. We didn't even think about using cryo-EM at the time because it's not possible, it's not there. So we actually started with crystallization. And then in 2016, we are kind of under discussion about, we should get a microscope. And successfully, by the end of 2016, we actually installed our own microscope. So, and then actually, once we have our microscope, so that's 1 year since we started the project, we got the microscope. Once we have our microscope, it was very fast to figure out the cryo-EM part. So in a few months, we were able to get our first structure. But then, as I explained, this protein is very difficult to purify, to stabilize the transmembrane domain. That took us about a year. And in the end, we learned different techniques like amphipose and nanodisk. And with the published structure, we put it into the nanodisk and different detergents. And so about 2 years, we were able to get that structure.
I was just curious which of these pockets you think, this Larexyl acetate inhibitors of TRPC6 that are reported are binding to.
Oh, what type of inhibitors? Sorry.
So they're these supposedly very potent Larexyl acetate-based inhibitors of TRPC6, and I was wondering if, which one of these pockets you think that will be relevant.
So far, the antagonist binding pocket, one is in the voltage sensor-like domain. We have one structure to show that it binds, and also we actually have another structure which is slightly different, but it's also this pocket. And then the other pocket is probably at the inner leaflet of the membrane bilayer, and that's the pocket that the other antagonist bound structure showed.
Are there any other questions for Yonghong? Oh, one in the back. Maybe we'll make this the last question. Thanks.
Very nice talk, and I can understand it's really tough work. So from structure, you suggested there is 3 site, agonist binding or antagonist binding site in this one, right, in TRPC. So all the trip channel are very similar in structure. What is your insight of other trip channel? Do you think that they will have the similar 3 binding sites, or?
So, over the past few years, there are so many trip channel structures coming out. And also, there are quite a few of them have the ligands. And I don't think I have read every paper, but the papers I have read, I try to summarize that. It seems that they fall into 3 different sites. So like in TRPC, like in our TRPC channel structures, we have found ligands in all 3 sites. And I think in TRPV channels, there are at least 2 sites that they find ligands. And in TRPMA channel, there are recent structures that identify 1 site. And because of the similar design of these channels, I would think that eventually you should be able to find ligands to modulate the activity on either of those 3 sites.
Oh, very quickly. Quick question.
From this structural, like 3 different sites, can you guess if any of the agonist binding site would be responsible for partial agonism, or one of the binding site would be responsible for full agonism?
Yeah, sorry. In terms of the mechanism, I think we still don't have enough structural information to answer.
Okay, great. Thanks for the questions. Thanks, Yonghong. Great talk. So it's my pleasure to introduce Professor Derek Bowie from McGill, also one of our scientific advisors for the meeting. So thanks for that.